Hello class, this is BIOT643, Techniques of Biotechnology, and this is lecture 1 on PCR, the polymerase chain reaction. Here's a brief outline of what we'll be discussing in this lecture. We're going to start off talking about basic PCR, take a look at some of the various types of PCR, look at PCR conditions in detail. We'll take a look at primer design, and then take a closer look at some of the different variations of PCR, namely quantitative PCR or qPCR, multiplex PCR, and droplet digital PCR or ddPCR. So first, let's take a look at the basic concepts and working of PCR. A typical PCR cycle has 3 different stages. We refer to these as denaturation, annealing, and extension. During denaturation, the template strands separate. These are the 2 strands of the double helix DNA, and so those become single-stranded. And this is the DNA melting process that you may have seen in lecture 0. The next stage is annealing. This is where the primers that are in the PCR reaction mix bind to their specific regions on the template strands. And then extension is where the DNA polymerase will build off of that primer, adding the new nucleotides downstream in the complementary manner to the template strand. And so these 3 stages get repeated multiple times, as we'll see in the next slide, and that causes an exponential increase in the number of copies of the targeted DNA sequence. So here's a slide that shows a typical PCR reaction diagrammatically. We have our double-stranded template DNA. So you see the 2 strands are bound together in their typical anti-parallel fashion. Here is essentially a typical PCR program, we would have our denaturation step, our annealing step, and the extension. These are the steps we just discussed. And you can see that they all occur at different temperatures and for different lengths of time. So the very first step, we enter the denaturation stage. As I mentioned, that is where the template DNA strands will come apart. And so here in this generic program, we have approximately 95 degrees or 94 for 1 minute, and that application of temperature helps the DNA strands to separate. After that, we lower the temperature down to, in this case, it looks like about 52. That is dependent on the DNA sequence of the primers, and we'll look at that in more detail later. But the general range is typically between 50 and 60, as you see in the diagram on the left. And so lowering the temperature down to that level allows the primers to bind. After that, we enter the extension stage. So after that, we raise the temperature, usually between 72 and 74 degrees. So that allows the polymerase to build the nucleotide chain off of the primers in the complementary manner to the template strand. And there we see that those amplicons have been completed, and we refer to them as amplicons, the products of the amplification. And then after that, we repeat the cycle again. So we see that we had initially started with one strand of DNA, and we now have two. So repeating the cycle, we would go back to denaturation. That would separate out these two strands, go through annealing, get more primers to bind, go to extension, build off the opposite strand. From there, we would end up with 4 different strands of target DNA, and then we'd repeat this multiple times until we get this exponential amplification. So this is a video on YouTube that shows the process quite excellently. It shows the extension off the primers, and in particular the exponential amplification. So I would encourage you to look at this video at your leisure. So let's look at some of the various different types of PCR. There are many variations of PCR. It is a highly customizable technique that can be modified to suit particular technical needs. I've listed in the next few slides a number of the different types of PCR. We won't be discussing all of them because that would take way longer than the lecture's already going to. So I've listed them for you to look at. Feel free to look them up and learn a little bit more about each of these. Some of the ones that we'll be discussing in the lectures, both to this lecture and the upcoming ones, are shown in red. So we'll be discussing cloning PCR. We'll be discussing multiplex PCR in this lecture, qPCR, and RT-PCR. So I mentioned the 3 different parts of the PCR cycle: denaturation, annealing, and extension. There are different PCR conditions that are involved in each of those steps, and these PCR conditions can determine whether a PCR is successful or not. So I find it worthwhile to take a close look at these different conditions and their effects. So I alluded to some of these conditions in the animated slide of PCR, but let's take a closer look at them now. So a typical PCR program, in addition to the 3-step cycle that is repeated, typically has an initial denaturation stage at the beginning. So in this particular case, we have a temperature of 95 degrees for 2 minutes. That can range anywhere between 1 minute to 5 minutes, again, or even shorter possibly. Again, depending on your particular situation and your needs. But that is typically only run once, hence the 1x. Here we see a denaturation, again at 95. That typically takes the same temperature as before for 30 seconds, an annealing at 60 for 30 seconds, and an extension at 72 for 30 seconds. And then this particular cycle gets repeated 30 different times. Denaturation, annealing, extension, denaturation, annealing, extension, and so on. And then at the end there's a final extension step to allow the polymerase to essentially finish all of the synthesis of strands that it's currently working on, and that's done for one repetition, and then the program ends. So as you can see, the two critical variables of all of these steps are the temperature and the time. So let's take a look at what roles and effects those have. These slides to some degree will refer back to some of the material covered in Lecture 0. If you watch these slides and they make perfect sense, great. If they're a little bit unclear to you, it may behoove you to go back to Lecture 0 and just review it briefly. But so looking at the background of denaturation, recall that the base pairs on the DNA strands are held together by hydrogen bonds, and those hydrogen bonds are weak, and applying thermal energy to the system will break the hydrogen bonds, and once those break, it causes the two strands to separate, and we call this denaturation or DNA melting. So during the denaturation step of the PCR cycle, we apply high thermal energy to break the hydrogen bonds that are between the two different DNA strands, holding them together. And the reason we need to do this is because the nitrogenous bases of the nucleotides are in between on the inside of the double helix. And so we need to separate out these strands in order for the DNA polymerase to be able to read those nucleotides and synthesize the opposite strand. So to help understand the process of denaturation, let's take a look at some of the potential problems we could encounter. An ideal temperature for the denaturation stage is typically considered between 94 and 96 degrees. Somewhere in that range. If the temperature is too low, the DNA strands won't be able to fully separate, and so what you'll end up with is partial separation, as in this diagram. And so if you recall, AT-rich regions have a lower TM, so if you have an overall DNA strand, it would start to come apart at the AT-rich regions first, and then sort of unzip in both directions until the entire DNA strand, both strands were separated. However, if the temperature is too low, the higher GC regions will stay bound together, and so you end up forming sort of these replication bubbles almost, where the nucleotides within the separated section can be read, but the rest of them aren't. And so if you look at this diagram, these are replication bubbles, but it does sort of show the same concept that I'm talking about. You have a section where the two DNA strands are separated at the arrows, and if your PCR target is located entirely within one of these sections, then you're fine. However, if it's in another section, such as the highlighted one here, where the two DNA strands are still together, then you will not be able to actually do your PCR off of that target, because the polymerase can't access the various sites. However, if your temperature is too high, say above 100 degrees Celsius, the individual DNA strands will start to break apart. And so recall that the hydrogen bonds are weak and will separate with the increase in thermal energy. However, high enough thermal energy can even break covalent bonds. And so even though the rest of the DNA backbone is covalently bonded, having too much energy or temperature in your PCR system can actually completely destroy the DNA strands. And the other potential downside of this is that because Taq polymerase is an enzyme, you can destroy its activity simply because the protein, the polymerase itself, will start to denature due to the high temperature, and that's a very unhappy Taq polymerase. Now let's take a look at the other parameter, time, with respect to denaturation. A typical denaturation step is approximately 30 to 60 seconds long. The problem with having too low of a time, say 10 to 15 seconds, is that you wouldn't give the two DNA strands enough time at the denaturation temperature in order to complete their separation. So here we see the DNA strands mostly bound together. As I mentioned in the previous slides, this denaturation process will begin at a high AT-rich region, simply because that has the lowest TM. The denaturation step will start there, and then unzip in either direction until that bubble gets larger. Simultaneously, you can have another bubble form in another AT-rich region, slightly lower AT-rich, which is why it's forming later. And those will continue to extend until they get larger and eventually meet and continue out towards the ends of the molecule until the two DNA strands have completely separated. But this process does take time, and so if you shortchange the denaturation step, you won't leave enough time for this to occur, and you'll end up possibly in one of these earlier diagram stages where part of the DNA is still bound together. And if that's where your target is, that's unfortunate from a PCR standpoint. So the problems that we see with too long of a denaturation step are exactly the same ones that we saw with too high a temperature. That is because, in fact, the time is not itself the critical parameter to these negative effects. It is too long of a time at the high temperature required for denaturation. And so if you extend the time of denaturation, assuming you have that properly high temperature, then you're just allowing extra time for those DNA strands to break apart and for the activity of the enzyme to be destroyed. So you want to reduce that time to only what is necessary. Okay, so now let's take a look at annealing. Recall that the TM is the temperature at which 50% of a given DNA sequence is denatured. During lecture 0, I described the DNA melting curves, and so this figure is just a refresher of that. And so you see the TM in this figure at 60 degrees. On the curve below the TM, you see most of the molecules in their double-stranded formation. whereas above the TM, they are in their single-stranded formation primarily. And so, yeah, that's exactly what I just said. So with that DNA melting curve in mind, the purpose of the annealing step is to reduce the thermal energy enough to allow some of the hydrogen bonds to reform, namely between the primer and the DNA strand, not the original opposite strand. And so choosing the proper annealing temperature is critical. You want the annealing temperature to be low enough to allow high sequence matches to form those hydrogen bonds, but high enough to break the weaker hydrogen bonds resulting from lower complementary matches. So let's take a look at that on this slide and discuss the thermodynamics of DNA annealing. We often describe primers as binding specifically to their region of complementarity on the DNA strand. However, sort of the dirty little secret of PCR and primer dynamics is that that actually doesn't happen the way we typically represent the process. Primers don't bind specifically. They can and do bind anywhere. So if, for example, we have this DNA sequence and we add primers to the mix, a primer will try to bind in this location, but it will also try to bind in this location and all the other locations in between and all around. So as it turns out, getting them to stay bound only to the specific target is what the selection of the proper annealing temperature will accomplish. So let's take a look at that. Here we have the primer trying to bind in this location, and you see only about half of the hydrogen bonds between the primer and the DNA strand of template are present. And so we end up with a bond strength, or a TM, essentially a measure of the bond strength of about 39.3 degrees Celsius. Over here, however, we have all the hydrogen bonds present because this is essentially the region that the primer was designed against. That's why it's a complete match. And so because all its hydrogen bonds are present, it does have a much higher TM, in this case 59.9 degrees. So recall that thermal energy is the same as temperature. So if we have a temperature of 30 degrees, we're essentially discussing 30 degrees worth of energy being put into the system. So for our purposes, I'm sort of equating energy and temperature almost equivalently. So we apply 30 degrees worth of energy into the system, which is unable to break the complete set of hydrogen bonds on either of these primer binding locations. And so those primers stay bound to each of those locations. If we increase the temperature to 50 degrees, now we're applying even more thermal energy into the system, 50 degrees worth of energy. That allows us to break the hydrogen bonds on this other location, and that primer is unable to bind, and therefore you will not be able to generate a PCR product off of that location. However, here at the location where this remaining primer is bound, not all of those hydrogen bonds were broken, so the primer stays bound and is able to form the primer that allows for the synthesis of the downstream DNA. If we apply even more energy into the system, we will be able to break the complete set of bonds from the primer on the left because we're applying 80 degrees worth of energy, and so that's greater than 39.3. However, that's also greater than 59.9 degrees. So we're able to break all of those bonds as well and neither of those primers is able to stay bound. And so because of that, if you were to choose very poorly an annealing temperature of 80 degrees, you would not, in this particular sequence and primer situation, be able to generate any PCR products. So now let's take a look at that in the potential problems format that we saw earlier. Typically we use a temperature of between 50 and 70 degrees for the annealing step that is dependent on what your DNA sequence for the primers is. So as we showed on the previous slide, if the temperature is too low, there is insufficient thermal energy to break the bonds between the primer and the DNA template sequence that is not an exact match, and that results in what we refer to as non-specific amplification. So you would be able to generate a PCR product from the primer on the right. That is the location that the primer was designed to bind to. So this is the good PCR product that you do want, but because the primer is bound here, you would also be able to generate a primer, or an amplicon from this location where the primer was not designed to bind to. So in this case, you would end up with two different PCR products on your gel, and because this is non-specific amplification, that is clearly something that you would not want, and your primers were not designed to do. However, if the temperature is too high, there would be too much energy. You would break the bonds between the primers and all the sequences, including those with an exact match, and you would have essentially no amplification because there would be no primers bound to your template DNA. So now let's take a look at the time parameter of annealing. Typically an annealing step lasts between 30 to 60 seconds. However, if the time is too low, you don't allow enough time for the primers to find the correct location on the template DNA and bind to it. So if you think about all the different primer molecules floating around in your PCR master mix, along with the template DNA, you have to allow enough time for the primers to find the location that they're designed to in this big floating mixture. And if you shortchange the annealing step in terms of time, you won't allow enough time for that to actually occur. However, if the time of annealing is too high, you end up with the opposite problem, which is allowing too much time for the primers to bind to the incorrect locations. However, as I mentioned on the slide, this really isn't a concern unless that's coupled with an annealing temperature that is also incorrect. And so here in the diagram, we see the same two primers that we saw previously. So if we were to, for example, extend this annealing step to, say, 5 minutes, which would be ridiculously long for a PCR, but a time of 5 minutes at the proper temperature, say 50 degrees, as we saw on the previous other slide, that would be able to, those 50 degrees worth of thermal energy would be able to break the bonds on the incorrectly bound primer on the left, but would leave in place the bonds on the correctly bound primer on the right. And so at 50 degrees you would only get your desired PCR amplicon. However, if we were to do 5 minutes at, say, 30 degrees, then both of these primers would stay bound, one to the correct location and one to the incorrect location, and you would end up with that non-specific amplification because you would get an amplicon from each of these primers and so you'd get 2 bands on your PCR electrophoresis gel. Okay, so now let's take a look at extension. Recall that the DNA polymerase creates a copy of the complementary template strand starting from a primer. And so this is one of the things that was covered in lecture 0. So if this seems a little bit foreign, I would encourage you to look at that. So here we see in blue the DNA template, and in red the short primer that is attached to it. The DNA polymerase comes along, attaches to the end of the 3' end of the primer, and will move down the DNA template strand, synthesizing based on the complementarity the opposite strand. And so the DNA polymerase only builds the strand in the 5' to 3' direction. So the purpose of the extension step is to raise the temperature to the optimal level for the activity of the DNA polymerase. It is an enzyme, and oftentimes enzymes have a particular temperature range at which they are most efficient. The reason that we use Taq DNA polymerase is because it is thermostable. It was isolated from the thermophilic bacterium Thermus aquaticus, and that's where the T and the AQ come from, and so we call that DNA polymerase Taq. It was discovered in Yellowstone National Park in some of the hot spring geysers, and because of the high temperatures of those geysers, this particular bacterium has evolved to be able to survive within that environment. And part of that survival evolution is obviously it needs to replicate its DNA, and so it needed to evolve a DNA polymerase capable of activity at such high temperatures. And so we humans were able to isolate that and figure out that it would be a really great enzyme to use in PCR reactions. Before we used Taq polymerase, we used regular DNA polymerases, but the problem was that that 95-degree denaturation temperature would destroy the activity of the enzyme. Most enzymes will not be able to function at such a high temperature. So if you look at the PCR diagram up the top, we would have denaturation, annealing, and then in the old days you would have to add the polymerase, and so it would do its extension. Then the PCR would cycle again, and you'd go back to denaturation, at which point the enzyme that you had added prior to the extension step is completely destroyed in terms of activity at such a high temperature of denaturation. So after the denaturation, you go back to annealing, and you need to add more enzyme in order to complete the next extension step. And so you would have to add enzyme prior to every extension step, which, as you can imagine, would be incredibly annoying to do 30 to 40 times. And so the discovery of Taq polymerase and its application towards PCR made a huge difference in the essentially automated way that these could be run, because the Taq polymerase could withstand the 95 degrees without losing its enzymatic activity. So now let's take a look at the potential problems with the temperature of the extension step. So Taq polymerase has a peak enzymatic activity typically around 72 degrees. Some of the different DNA polymerases will have slightly different activities, but typically it's in that same range. However, that is a spectrum, and so you can see on the graph here, in yellow we have TAC, and in red we have a different enzyme called PFU. Let's just look at TAC for now. So right around 70 degrees ish on this graph is the peak level of activity. However, as you can see on either side of that level, you still have some activity, but it's not at its most efficient. And so whether the temperature of extension is too high or too low, you're losing your extension and therefore amplification efficiency. So it's good to understand the efficiency profile versus temperature of your particular enzyme. And typically when you order an enzyme from the manufacturer, they let you know at about what temperature you should run your reactions. So the time of extension can vary based on your desired PCR target. Typically it'll be in the range of between 30 seconds to 1 minute, depending on the size amplicon that you're expecting. It can range up to as high as 5 minutes or even longer. So if the time that you provide for the extension step is too low, that TAC polymerase, or whichever polymerase you're using, doesn't have enough time to synthesize enough new DNA before the next denaturation cycle begins. So we can see that diagrammatically here. We've got our DNA template as we saw before. We have our primer in red. We have the DNA polymerase come and bind to the end of the primer and begin its extension at the 72 degree extension temperature. So it starts making its DNA, but say we cut this step a little too short. Now the PCR program is going to go back up to 95 degrees, and that is going to denature the strands, which is going to cause the polymerase to detach, because it will no longer have the template strand in order to read the base pairs, so it'll know which ones to add to the new strand. And because that polymerase detached when the strands separated, now you have this entire region of unsynthesized DNA. And so if that was part of your desired target, shortchanging the extension step in terms of time will result in poor amplification, or at least incomplete amplification. So there is a generally accepted parameter of 1 minute per kb, that's kilobase pair, or 1,000 base pairs. That is a very good kind of ballpark figure. So if you have, for example, a 2,000 base pair fragment that you want to amplify, you would want to allow for an extension time of 2 minutes because you have 2 kb. So now let's take a look at primer design. So I like to think of the primers as one of the most critical parts of the PCR reaction. In reality, all of the components are equally critical because your PCR reaction won't work without all of the components in the mix. You need the polymerase, of course. The NTPs are the building blocks of the newly synthesized strand. The polymerase requires magnesium, and of course you're building off the 3' OH of the primers. So it won't work unless all of those components are there. But in terms of the specificity and where you're going to be amplifying from, the primers is really the prime determinant of all of those different parameters. So here we have listed the primers are responsible for determining the target sequence that will be amplified. And so the forward and reverse primers will bind to their specific locations and amplify the sequence in between the two primers. If the primers do determine the sensitivity and specificity of the amplification reaction, the length of the amplicon based on where those primers bound, and also the annealing temperature of the PCR reaction. And so fortunately, primers are one of the most controllable and customizable parameters that you have control over. And so I always like to highlight for my students that proper primer design is the single biggest factor in your control that will influence the success or failure of a PCR. So it's important to understand what good primers look like, what potential problems you could have, and how to design a good set of primers for your specific reaction. So let's take a closer look at that. So that last slide probably made it seem as though it was going to be a cut and dry, straightforward process. That is to some degree true, but unfortunately I have found that primer design is much more of an art than a science. The reason is that there are a huge number of factors. They're not entirely independent, and so they're interrelated with each other, and you're trying to juggle all four of these particular factors and some additional ones all at the same time. And if you for example, increase the length, that will also increase the GC content potentially, or decrease it. And so you're juggling a number of different balls in the air, trying to make them all stay up and end up with a good PCR reaction. And so that said, there are some generally good guidelines for making good primers. Typically, primers are designed to be between 18 to 22 base pairs. They can be a little bit shorter or longer as necessary, but about 20 tends to be the target that people shoot for. The GC content is about half, anywhere in there. And if you recall from the DNA melting curves, increasing the GC content will increase the TM. So that is how those two are interrelated. So with that, we shoot for a TM of between 40 to 60 degrees typically, and that is probably the parameter that's most sequence dependent on exactly what it is you're trying to amplify. And then you do have two different primers, the forward and reverse primer, and each of those primers has its own TM, and you want the difference in TM between those two primers, what we call the delta TM, to be less than 5 degrees typically, and we'll get into each of these parameters at length to determine what some of the potential problems with those are. So let's start off looking at the length parameter. The length you choose for your primers should be enough to allow for sequence specificity within a sample. So what do we mean by that? Let's imagine that the DNA that we're trying to amplify from is distributed randomly. So each of the nucleotides A, T, C, or G have an equal chance of occurring, so that'd be 25% for each. In reality, of course, DNA is not distributed randomly, especially at local sections. You might have high AT rich regions in some areas and high GC regions in other areas. But for the purposes of this discussion, to understand the length parameter, let's imagine that all the DNA is distributed randomly. So how many combinations of random sequences are there? The number of combinations is 4 to the n, where n is the number of nucleotides in length of your given sequence. So let's take a look at that. Let's say that we have a 1 base pair sequence. Obviously, the 4 options for a 1 base pair sequence is a sequence of A, a sequence of T, of G, or of C. And that's it. Those are the only 4 options to choose from. If we increase our length to 2 nucleotides, you have 16 combinations. So you can have an AA, an AT, an AC, an AG, a TA, a TT, and so on. And so this is simply 4 to the second power, or 4 times 4, which is 16. If we increase that length to 3 nucleotides, now we're looking at a combination number of 64. You can have AAA, AAT, AAC, and so on until you complete all of the 3 nucleotide combinations. You'll find that there are 64 of those. Okay, so those combinations are interesting, but what relevance does that have for primer design, and in particular, what are the implications for sequence specificity? Well, let's start off by looking at the first example that we had on the previous slide, which was a primer length of 1 nucleotide. As I said, with a primer length of 1, you have 4 different options, A, T, C, or G. And so if we imagine in our idealized E. coli genome that just happens to be unrealistically completely randomly distributed, you would have an equal number of A's and C's and T's and G's. So the frequency at which any given nucleotide in this case would occur would be the entire length of the genome, which is the 4.6 megabase pairs that you see on there, divided by the number of combinations, which in this case is 4. And so if you pick a letter, let's say A, you would expect to find 1.1 million different A's within the randomly distributed E. coli genome. So you don't want to have that many places that your primers can bind. You want them to be far more specific than that. So if we look at the second example that we had on the previous slide, where we have a primer length of 2, you have 16 different combinations, and so you would divide the length of the target genome by the number of combinations. So again, if it was randomly distributed, if you pick one of the combinations, say TA, you would expect to find within the genome the letter T followed by the letter A 289,000 times, and so that isn't terribly specific either. So with 3, we would be doing the same thing. You'd end up with any given 3 nucleotide sequence distributed 72,000 times within the E. coli genome. So we can expand that out further, again just using that formula of 4 to the n, where n is the primer length. And so you see for a primer length of 4, you'd have 256 combinations, which would occur 18,000 times, and so on. And so you see right around 11 base pairs or nucleotides for the primer length is about the point where you sort of break even and only have that one specific location. But you actually want to be even more specific than that, if at all possible. And so you see by a length of 18, we're down to 0.0001 occurrences per E. coli genome. And again, this is randomly distributed. So obviously, if that distribution was in reality, you wouldn't have that particular sequence site, and so your primer wouldn't bind anywhere. But when you design your primers, you know the site sequence that you're binding to, and you design it against that particular sequence. So when you design your primers, if they are long enough, you should have reasonable guarantee that that site that you're looking at is the only one within the genome. And so typically primers are around 20 nucleotides in length. And I don't have 20 here on the on this particular slide, but 4 to the 20 combinations means it's very, very unlikely to actually occur, so that you can essentially be very confident that your particular site is the only one your primer will bind to, and you won't have another situation like that at random, and you shouldn't end up with non-specific amplification. Okay, so now let's take a look at GC content. As I mentioned previously, the GC content you typically want to be between 40 to 60%. And the reason for that is that an AT base pair has 2 hydrogen bonds, while a GC base pair has 3 hydrogen bonds. So you need more energy, in the case of PCR thermal energy, to break a GC bond than you do an AT bond. And let's take a look at why that matters for primer design. Let's imagine this sequence here is the one that we're trying to amplify from. If we tried to bind our primer here, that particular target sequence is 50% AT and so 50% GC. And that gives us a TM of 53.2. If we take the same sequence and just move the primer over just a few base pairs, then now this particular sequence is much higher in CG, and so you end up with a much higher TM. It's almost 10 degrees higher. And so that could have definite amplification effects, and so you would want to take those into consideration. Okay, so now let's take a look at TM and TA. We've discussed the TM of your individual primers. The TA is the temperature of annealing of your PCR reaction, and so that would be what that middle step in your PCR program, the annealing step, what temperature that's set to. So generally you want your primers to be designed with the lowest TM that will allow specific binding only to your desired target. So if you think back to slide 23, the thermodynamics slide, the primers bind to all of the DNA everywhere. What determines whether they stay bound is the TM of that particular binding at that site, as well as the amount of energy you're putting into the system, which in this case is the TA. That's the temperature of that annealing step. And so the general guideline is that you want your annealing step temperature to be 5 to 10 degrees lower than the TM of your primers. And let's take a look at the DNA melting curve to sort of visualize why that's the case. So recall that the TM is where 50% of the single-stranded or molecules are in their single-stranded formations, and so here they've been denatured So we see our example 10 strands of DNA, 5 of which are single-stranded and 5 are still double-stranded. So the double-stranded ones would represent the template DNA with a primer bound to it, whereas the single-stranded ones would be primers that can't bind to their specific locations. But if we take a look lower on the curve, so this DNA curve has a TM of 60. If we look at 55 degrees, we have a much lower, it's about 0% single-stranded frequency. And so we see all the molecules here being double-stranded. That is to represent the primers being bound and able to stay bound. And of course, if you remember the way the DNA melting curve works, if we go above the TM, now we're breaking more of the bonds and approaching the point where none of the primers can bind to their respective locations. Okay, now let's take a look at TM mismatch. So you typically want your delta TM to be less than 5 degrees. Let's take a look at why you want that. So with this example sequence that we have here, we have our forward primer binding in this location, and we have our reverse primer binding in this location. So if we take a look at the TMs of this sequence, the forward primer has a TM of about 50 degrees. The reverse primer has a much higher TM of about 65 degrees. So why does this matter? What are the implications of this? Let's take a look at the DNA melting curve to understand why this is important. So let's first look at the graph of the forward primer. You see the TM is right about 53 degrees. So that's where half of its particular primers are not bound to the template sequence. If we plot the reverse primer on the same graph, we have its TM at 65 degrees. So what's the big deal? Why does that matter? Well, if you were to run your reaction at the TM of the reverse primer, 50% of the reverse primer being bound means that essentially none of the forward primer has bound, because all of those molecules are in their single-stranded formations. So none of those primers can bind, which means that you won't be getting your amplification expansion, that exponential amplification that we've seen. However, if we take a look at the TM of the forward primer, where 50% of its molecules are bound, now we have essentially all of the reverse primer bound to various locations along the DNA sequence. And so there isn't enough energy to break some of the non-specific binding that the reverse primer will attempt while those reaction conditions or the delta TM is so high. So let's take a look at ways that we can address a delta TM. If you have flexibility in the positioning of the primers, you can simply move the primers. And if for some reason you don't have that sort of flexibility, you can actually change the primer length. So let's take a look at those situations. Okay, let's start off looking at changing the primer location as the first method of reducing your delta TM. Here we have the DNA sequence, the primers, and the DNA melting curves that we saw 2 slides ago. So instead of having the forward primer bind where we have it now, let's try putting it a little bit closer. So we move it up just a couple base pairs, and here this primer has a TM of 62.3 degrees, so that's a delta TM of 2.7. So if we erase that other curve from the previous primer and plot this one, now the two curves are much more closely aligned. So what does that mean? That means that even at 50% at the TM of the forward primer, you still have, in this case, about 15% of the reverse primer bound. Not ideal by any means, but definitely workable. This would most likely result in a working PCR. And here we have essentially the opposite situation, where 85% of the forward primer is single-stranded, so 15% of the forward primer is bound at the TM of the reverse primer. So again, not terribly efficient, but definitely much more workable than the previous situation, where you had essentially none of the primers at either TM being compatible with the other primer. So now let's take a look at the other method of decreasing the delta TM, which is changing the primer length. So say we have this example DNA strand, and we have a 300 base pair sequence in the middle that we want to amplify. And so we design our primers to sit immediately outside our region of interest. And so this is 18 base pairs outside, and this primer has a TM of 61.8 degrees, and we would design similarly our forward primer to sit immediately outside our region of interest. And so the lengths will be the same. We designed this at 18 base pairs. But as it turns out, the TM of that primer is 50.2 degrees, and that is far too great a delta TM for efficient amplification. So let's take a look at what the effects of changing that would be. So in the current situation, we would have an amplicon of 336 base pairs. Keep in mind that the PCR product, the amplicon, would be the 300 base pairs in the middle, but then it would also include the DNA that comprises both of the primers. So 300 plus 18 is 336. So that's where that number comes from, because the primers do become part of the final amplicons. So if we were to extend the length of the primer a little bit further back, so you see this newest version of the primer sits in exactly the same 18 base pairs, but just extends back a little bit further. So with those additional nucleotides, once you take those into account, the length of the primer is now 24 base pairs, and because we have additional hydrogen bonds as a result of the increased length, now the TM of that is 56.3. Because of the additional length of the primer, we do add a little bit more to the final amplicon. So we extended the primer itself by 6 base pairs, which means that the final amplicon is also 6 base pairs longer. So this TM is certainly more compatible with that of the reverse primer in this situation. So let's go out just one base pair further. Now we're looking at 25 base pairs and a TM of 56.9. 1 degree further, or I'm sorry, 1 base pair further increases it even more. And then once we get to, in this case, 27 base pairs, we're looking at a TM of 60.7 degrees, and that's just about 1 degree difference from the reverse primer. So of course this is just all example sequence. The amount that it would change relative to the length is based on what the sequence actually is at those locations, but this gives you one more tool in your arsenal for reducing the delta TM. If you don't have the flexibility to move your primers, you can just extend them out a little bit. And so in this particular example, I increased the length of the forward primer. If, for example, we wanted to, instead of bringing the forward primer's TM up to that of the reverse primer's TM, we could try to bring the TM of the reverse primer down to that of the forward primer. In this case, that would entail shortening the reverse primer. In this particular example case, that's not too much of an option because the primer length is 18 base pairs, so it's already a little bit on the short side. I wouldn't want to go much further short than that. You could potentially lower this to 17 or 16 nucleotides in length, but doing that potentially means that you could result in non-specific amplification because the primer could bind to other locations, and so that's sort of the offset. It's kind of that juggling act that I talked about between the various parameters. So in this particular example case, it would be better to increase the TM of the forward primer than to try to reduce the TM of the reverse primer. But in reality, you can have the opposite situation. And so, of course, it's all dependent on the actual sequence. So this is a good diagram showing some of the potential results from various primer designs. In the first lane of the gel, we see an amplicon generated from specific primer binding to the location of interest of the target DNA. In the second lane, you see no amplicon. That suggests that one or both of the primers weren't able to bind and stay bound to the DNA template. That could also be a factor of the annealing temperature, and so that harkens back to the thermodynamics of the TA and the TM. In the third lane, we see a product, but it is of the wrong size, so that suggests that one or both of the primers weren't binding to the right location, but were able to bind and generate a product. And in the fourth lane, we see 3 different bands, one of which is the correct size, and the other 2 that aren't. And this suggests that in addition to the correct amplicon being generated, which is the middle one of the 3, the primers also bind to other locations non-specifically, and cause a full amplification to occur. and so you end up seeing those on your gel. And that's a sort of non-specific amplification that you can sort of winnow down with some of these various primer design tricks. Okay, the last important concept of primer design that I want to cover is secondary structures in your primers. So it's important to realize that primers will bind to their complementary sequences wherever they are. They don't care where it is. And the implications of this can manifest in 3 different types of secondary structures: hairpins, self-dimers, and cross-dimers. So let's take a look at each of these individually. A hairpin is quite simply where a primer binds to itself. So say, for example, we have this 20 base pair primer, and we're hoping that it will anneal somewhere that we want it to in our template DNA. That might happen. However, the problem with this particular primer is that there is some complementarity within the primer itself. So if we take a look at these regions right here, reading from 5' to 3', G, G, A, C, and then some sequence in between, and then G, T, C, C. If you read it strictly 5' to 3', there doesn't seem to be too much of an issue. However, if this molecule were to actually bend in on itself, you can see now that you have complementarity in the anti-parallel fashion. So even though the molecule overall is 5' to 3' from the blunt end of the arrow to the pointy end of the arrow, you can imagine that the top strand is 5' to 3', and the orientation of the bottom half of that strand is 5' to 3' from the left. From the right. So 3' to 5' from the left. So these are now anti-parallel, which means that you can form hydrogen bonds between the G and the C, the G and the C, the A and the T, and the C and the G. And so the fact that these hydrogen bonds occur in the same molecule is irrelevant to the hydrogen bonding process. As I said, the molecule itself doesn't care. It's simply going to form hydrogen bonds with a sequence that it finds to be complementary. And so this molecule would actually bend in on itself like this. So what's wrong with that? Well, quite simply, the fact that those primer molecules are bent in on themselves and hydrogen bonded means that there are much fewer number of molecules available to bind to the template DNA. And so the net effect is to dramatically reduce the concentration of primers available to initiate that amplification. So the result is poor amplification that manifests in very faint bands on your gel. A similar situation occurs when we have self-dimers. This is where 2 copies of the same primer bind to each other rather than a single molecule bending in on itself. So if we have, for example, this 24 base pair primer molecule, let's call this our forward primer, a second copy of the forward primer will have the same sequence, but if you orient it in the anti-parallel fashion, and there happen to be these regions that will form complementary base pairs in this, when oriented in this manner, you can get these primers binding to each other. Again, the fact that it's on another copy of the same molecule is irrelevant to the binding process. Each of these primers sees the opposite base pairs that it's looking for and forms bonds. And what this will result in is what we call primer dimers. So the reason for that is that the polymerase will come along and just as though the PCR reaction were occurring normally, it will slide down in the direction of the template strand, which in this case just happens to be another copy of the same primer, and we'll put down the complementary base pairs, and another copy of the polymerase will do the same thing on the other strands. And now instead of two 24 base pair primers, you have a amplicon, very short amplicon of 37 base pairs, which is accidental in nature because this was the primer dimer and not your desired product. The primary effect of this is to reduce the number of primer molecules that are available to bind to the DNA template, just the same way that we saw with the hairpin primers. And so the main manifestation of this is poor amplification, and that would be observed as faint bands on your PCR gel. The other way you can tell that you have primer dimers forming is with the low molecular weight bands that you would see at the bottom of your gel. So here is an example of a gel with varying concentrations of primer dimers being formed. Down here, as I said, they're very low molecular weight, so they're typically show up below all the bands on your ladder, and as you can see, the amount of primer dimer is sort of inversely proportional to the amplification of your desired product. So here we're on this lane where we have a nice big bright band. We have very little primer dimer showing up. However, over in this lane where we have a lot of primer dimer forming, that takes its toll on the desired amplification. And so if you think about that, the number of primer molecules that are incorporating into the eventual primer dimers, that just means that that's that many fewer molecules that are available to initiate the PCR amplification from your desired target. So overall, primer dimers are bad. The situation with cross dimers looks a lot like what we just saw with the self dimers, except that instead of two copies of the same primer molecule, you have two completely different primer molecules that are binding to each other. So say for example we have a forward primer with this sequence, and we try to amplify with a reverse primer of this sequence. Each of these primers are designed to bind to a specific part of the template DNA, X length apart, say 300 base pairs, and amplify that. However, despite the fact that they are designed to sit 300 base pairs apart, it also turns out that there is some complementarity between each of these primer sequences, so they can end up binding to themselves, and then as we saw with the self dimers, the polymerase would come along and extend the rest of those strands until you had the complete primer dimer. so the manifestations and effects of this are essentially the same as we saw with the self-dimers. Low molecular weight primer dimer bands at the bottom of your gel, and poor amplification of your desired products. Okay, so that was all the bad news, all the different ways that you can mess up primer design with respect to secondary structures. The good news about that is that it's actually pretty easy to avoid. Most of the software that's used in primer design will also analyze the structures that could potentially form based on the sequences of your primers. So they'll look for hairpins, they'll look for self-dimers, they'll look for cross-dimers, and let you know if there's a potential problem, and usually suggest some alternatives also that may be less thermodynamically likely to form such structures. Okay, well, now that we've taken a very close look at PCR, including all the various conditions involved, the various problems that can occur, all the thermodynamics, and good primer design, let's take a look at some of the various applications of PCR and the variants of the process, starting with quantitative PCR or qPCR. qPCR is also known as real-time PCR. However, it is imperative that you not abbreviate that as RT-PCR, as that is the acronym for reverse transcriptase PCR. These are two very different PCR techniques, and so definitely don't call it RT-PCR. Anybody who submits an assignment calling real-time PCR RT-PCR may or may not lose a few points on that. Probably not, but why take the chance? I encourage you to refer to this as qPCR. So, okay, in all seriousness, qPCR incorporates a fluorescent probe into the PCR mixture. It's called real-time PCR because at each stage of the PCR amplification, the level of fluorescence in the PCR tube is detected in red. And because this is occurring at each cycle of the amplification, it's different in the way that the results are read compared to endpoint PCR, which is where the entire PCR protocol has to be run to completion before you can detect the results. So when we say endpoint PCR, we call it that because you do have to run the entire protocol. So that would be your standard PCR, where you would then open up the tube and aliquot some of your PCR mixture in the tube where the amplification occurred. You would load that into a gel and then examine that via electrophoresis. With qPCR, you don't have to do that. It detects the fluorescent level, which is correlated with the amplification at each cycle. And here's a table comparing standard PCR with qPCR, and some of this is what I just discussed. So standard PCR uses endpoint detection, whereas qPCR uses real-time detection. In standard PCR, you do have to remove part of the sample from the PCR tube and load it into a gel for electrophoresis. That means that you could potentially contaminate some of your other PCR reactions and so on. Whereas with qPCR, there is no electrophoresis that's required. The fluorescence signal is actually read through the PCR tube, which means you don't need to open it, and so there's no post-PCR manipulation. which dramatically cuts down the possibility of contamination with PCR products. And then PCR, the standard version, is considered to be either qualitative or at very best semi-quantitative, because the band intensity does correlate with the level of amplification. So the brighter the band, the more template DNA you had, resulting in a greater amount of amplicon at the end. Whereas with qPCR, the Q means quantitative, and so when you plot the individual data points in terms of fluorescence, that allows the concentration of the original template DNA to be calculated, and we'll take a look at that in just a bit here. So how exactly is the amplification from qPCR detected? There are two main methods for doing this. The first is the probe-based method. One example product of that is called TACMAN. The next is the intercalating dye method. An example product of that is called CyberGreen. Let's first look at the TACMAN method. So if you look in the diagram on the right, at the top part, the number 1 polymerization part of the diagram, you see your forward primer on the left, your reverse primer on the right, and then you have this third molecule called the probe. It hybridizes to the DNA. Essentially, it's almost like a third primer for your PCR reaction. On one side of the probe is your reporter. In this diagram, that's shown by the orange R. On the other side of the molecule is the quencher, in this diagram shown as a green Q. And the reporter molecule fluoresces at a specific wavelength that can be detected by the qPCR machine. However, that same wavelength is absorbed by the quencher, and so it's fluorescing at one wavelength, and the quencher is absorbing all the light emitted at that wavelength. And so what you end up with is not being able to detect any fluorescence when the quencher is in proximity to the reporter, and so when they're on the same molecule of the probe, there's no fluorescent signal detected. So in step 2, where the strand displaces, the polymerization of the reaction begins. And so you see now the arrow coming off the forward primer is bumping up against the probe and beginning to displace it. Once that reaches the part where the probe has been hybridized, it will begin to cleave off that part of the molecule. And so as you can see in number 3, the cleavage section, that reporter molecule has now been displaced off of the probe, and so what we end up with, that reporter floats off into the reaction mixture because it's no longer in such close proximity to the quencher. Now it continues to fluoresce as it was before, but that fluorescence is no longer being entirely absorbed by the quencher and can therefore be detected. And so now, with the help of the qPCR imaging software and analysis software, we can detect at this cycle that particular molecule is now fluorescing, and therefore you know that amplification did occur up at that point. And so then when the next cycle of PCR begins, you'll have your primers anneal again at the annealing stage, and you'll also have your probe molecule anneal, and you'll repeat this process over and over until all the reporter molecules have been cleaved off of the probes, and you can monitor that at the various cycles, and we'll take a look at the graph of that in a couple slides here. So the other method is the intercalating dye method, and as I mentioned, cybergreen is probably the most commonly used intercalating dye for qPCR. So cybergreen will fluoresce when it is bound to double-stranded DNA, and the word intercalating means that it actually fits in between the two different strands of the DNA. So you can see in number 1, the top step, the cybergreen molecules are in between the two different strands. When the DNA is denatured, those cybergreen molecules are no longer between the two strands because the two strands are not together, and so that level of fluorescence is reduced. When the polymerization occurs and the two molecules have the two strands have been reconstituted together, the cybergreen can fit again in between the two strands and begin to fluoresce again. And so we can monitor the increase and decrease of the cybergreen fluorescence at the various stages, and based on how much double-stranded product there is, we can plot that increase and therefore plot the amplification. As the qPCR reaction progresses, the fluorescence will reach a threshold level that can be detected. The fluorescence is measured in RFUs, or relative fluorescence units. That level of fluorescence is recorded at each cycle. Now, if you recall the previous slide with the cybergreen, we saw that even at the denatured stage, there is some level of fluorescence. It is drastically reduced relative to the intercalated stage where the two strands are bound together. But at that denatured stage, you still do see some fluorescence, which means that at some point there is a baseline fluorescence, which is kind of the lowest level, but it will never be zero. And so the point at which that fluorescence exceeds the threshold is referred to as the CT cycle. So if you look at the graph on the right, you see that orange line down at the bottom, that is the baseline level of fluorescence. And so this particular graph, if you look on the left axis, says PCR baseline subtracted RFU. So this graph has been normalized to subtract out that baseline fluorescence and really only report the results above that baseline. So you see the curves. There are some replicates in there. As you can see, there's several different colored lines that kind of line up all together. And there's a lot that goes into reading one of these qPCR readouts, and we won't get into all of that, but I just want to introduce you to some of this. So the way that this is typically done is to do several replicates just to make sure that your different curves are coming out very tightly and showing a lot of reproducibility. But then you also will typically dilute your DNA by tenths, your template DNA, and so that results in different cycle numbers where the threshold will be exceeded. So different concentrations of starting material will result in different CT values. More template will result in a lower CT value because the threshold is reached sooner. And so if you think about, let's say, this first green curve here, let's see if I can use a laser pointer. So this green curve here, the CT or cycle threshold is at 14 cycles. So after the first 14 cycles of PCR have been completed, that's where the level of fluorescence begins to be detected. So if we were to take that DNA and dilute it, say, by 10, so do a 1 to 10 dilution and run that as another sample, we would increase the CT value to, it looks like, about 17.5 or so. And so you see a couple replicates there. If we were to dilute that out another 10, so 100-fold relative to the initial sample, then we increase the CT value there, and again you see these nice evenly spaced different dilutions. So if we take the results from the graph on the left, which is similar to the one we saw on the previous slide, and plot those on a linear log graph, as opposed to log linear, such as the one we have on the right, we can plot them and establish a linear standard curve that allows us to back calculate the amount of DNA in the original template solution, and this also helps to plot the repeatability between the replicate reactions to see how consistent they are, and this is where the absolute quantification part of the qPCR assay is very valuable. So now let's look at multiplex PCR. In multiplex PCR, you are running a single PCR reaction. That is to say, you have a single tube with a single PCR reaction mixture in there, to which you've added your template DNA. However, rather than just one primer pair, you add multiple primer pairs. These primers will bind to different targets within your template DNA and create different amplicons. So for example, if we have a plasmid or a small chromosome, we can add primers to amplify from this location, and we'll generate this particular product shown in red. But we can also add two other pairs of primers, which will generate these two differently colored bands. And so because you are doing this in a single tube, you only have one reaction volume of reagents, so you don't need to run one tube for the red reaction, one for the green, and one for the blue. You're only adding one reaction volume worth of template DNA. That can be particularly important when your DNA is very precious. When DNA is extracted from clinical samples or patient biopsies, anything like that, where you're working with very limited quantities of DNA. You want to eliminate the number of reactions that you run and reduce those down to a minimum, and multiplex reactions can definitely help with that. You have one reaction time, so you don't need to run the red reaction and then later run the green reaction and then the blue reaction again, and you can run these all out on the same gel. So for example, because these would all be coming out of the same tube, you can load the PCR products into the well of your gel, and when you run them, you'll end up with all three products in the same lane, separated out by their different sizes. And so that's one of the factors that you need to consider. You have to make sure that your amplicons are different enough in size to be resolved as separate bands on your gel. You do have to think back to the various primer design concepts that we discussed. So in this particular diagram, you have 6 different primers. You would want them to all have approximately the same TMs and not be different. And you simply have more primers in the mix, and that allows the potential for additional primer dimers to form. Because recall when we were looking at the various secondary structures, we were looking at the hairpins, the self-dimers, and the cross-dimers. The hairpins wouldn't really change because that is, it bending in on itself. The self-dimers, again, you'd be looking at the same other copies of the same primer, but with the cross-dimers, you have much more potential in multiplex PCR to get cross-dimer formation, because now you don't have just a single pair. For example, if we're talking about the red primers, we're not just worried about the red forward binding to the red reverse. We're also worried about the red forward binding to the green forward or the green reverse, or the blue forward or the blue reverse, and then all the various combinations of this. And so some of the software is also able to determine whether there will be problems with multiplex primers as well. So here's a real-world application of multiplex PCR. This is a multiplex PCR to detect and characterize Bacillus anthracis. Bacillus anthracis is the causative agent of the disease anthrax. It is a gram-positive rod that has a single circular chromosome and 2 circular plasmids, PXO1 and PXO2, which encode the toxin and capsule genes for anthrax, respectively. Here is a diagram of the readout from this multiplex PCR on a gel electrophoresis. The way this PCR is set up is to amplify 2 targets from the chromosome, 4 targets from PXO1, and 3 targets from PXO2. So here you see, for example, the 2 spots on the chromosome where the targets are for the primers in this multiplex PCR. And so one of these amplicons is 555 base pairs. That's part of the 16S gene. The other is an SSPE gene that is specific to Bacillus anthracis. That's 188 base pairs in length. And so you have one being the top band, the other being the bottom band. Then we have 4 targets on PX01. These are the 3 toxin genes and 1 control gene. They all have different amplicon lengths. And the 3 targets on PX02, 2 of them being the capsule genes, 1 being another control gene, all have different amplicon lengths as well. And so you end up with this nice 9 base pair. I'm sorry, not 9 base pair. 9 amplicon sort of distribution of the different products. And so you're able to tell whether it has PX01, whether it has PX02, and whether it is Bacillus anthracis. And this is a PCR that I designed at work at my main job. And I used a lot of the techniques that we discussed in terms of primer design and changing annealing temperatures and so on. And so there's a lot of work in particular that goes into designing a multiplex PCR, because if you change one of the amplicon sizes, it may get too close to one of the other amplicons to be able to differentiate on a gel. And so there's a lot that goes into this, and I personally think it turned out pretty well. There's a citation down at the bottom of the slide if anybody's interested in reading up on it. I'd be happy to explain it if anybody has any questions, but I think because of the way that this groups the different amplicons, it shows the overall concept of multiplex PCR fairly well. So now let's talk about one of the newest variations of PCR. This is droplet digital PCR, or ddPCR. The hallmark of ddPCR is the massive partitioning of the sample that is accomplished through water, oil, emulsion, droplet, and microfluidics technologies. And so the procedure that is used is that a sample containing the PCR reagents and template DNA is mixed with oil. Then this is fractionated into many droplets, usually on the order of 20,000. And each of these droplets acts as a separate PCR reaction. And if the template DNA is present in that particular droplet, it will be amplified, and then that can be detected. So the generation of droplets for ddPCR is accomplished using a device creatively referred to as a droplet generator. This slide will sort of summarize the inner workings of the droplet generator. So you start off with a tube containing your PCR mix with your template DNA. And for those who may have downloaded the slide presentation before this lecture was recorded, I had labeled that tube initially with just template DNA. That is incorrect. It should be PCR mix plus template DNA, which is why I've highlighted that in red on this particular slide. So the tube containing your PCR mix and template DNA feeds into this microfluidic chamber, and so the mix gets sucked up and inserted into the microfluidic chamber, where it is then mixed with the emulsion oil that causes a droplet to form of oil surrounded by the PCR mix containing the template DNA, and then that droplet is pushed into the droplet chamber, and the process is repeated numerous times for many droplets to form. After that, all those droplets are collected Some of them will contain PCR mix with template DNA. The others may contain only PCR mix. That depends on the concentration of your template DNA within the PCR tube. The next step is the PCR amplification stage. This is essentially the same as a standard PCR reaction. The only difference is that instead of a single tube containing your entire mixture, these emulsified droplets are added to the tubes, usually in a nice 6-well plate format. And these droplets actually stay separated, and so the droplets don't recombine into a single volume. They stay fractionated in this droplet form. After the PCR amplification has completed, the samples are transferred to an optical reader. There, the droplets are channeled through a microfluidic chamber, and you see a picture of that down at the bottom. In that chamber, the individual droplets are squeezed through that narrow microfluidic channel, where they're illuminated by a laser, which can detect the level of fluorescence in each individual droplet, and those droplets are read very rapidly. And you see an example of the readout from those droplets, showing whether there was amplification or not in the circular diagram on the right of the slide. So here's a link to a video from the company BioRad on their ddPCR technology. It's an excellent video that describes the process well, and I would encourage you to look at that. So as I mentioned in the first slide of this section, the key to ddPCR is the massive partitioning of the sample. So if you look at the standard PCR diagram on the left, you have essentially one giant single reaction from which you can only obtain one measurement, namely whether the DNA was there or not, based on the level of amplification. Whereas with ddPCR, each of these oil droplets is an independent single amplification event, and so you can take that exact same volume and divide it into many thousands of discrete measurements, and you can use that to calculate the concentration of DNA in your original sample as well. So here's a slide again from BioRad on where ddPCR fits into the history and progression of PCR. The first generation of standard PCR, the endpoint PCR that we were discussing, is qualitative, and so the best you can do is the band there, how bright is the band, etc. Then the essentially second generation of PCR was the real-time PCR, where we were able to calculate the relative quantification of the template sample and detect it in real time, the amplification. And then we have ddPCR as the third generation of PCR, and this allows us to quantitate absolutely the template DNA concentration that we have in the sample through this massive partitioning. So here are the references for this lecture. If you have any questions on any of the material discussed in this lecture or on PCR in general, feel free to send me an email or post on the website. I'd love to help you understand PCR better. It is one of my absolute favorite subjects, and I really enjoy talking about PCR. Okay, thanks. Have a great week.